Hello friends, welcome to Biosumit. Today we learn about ion exchange chromatography. So this is the new video of the chromatography series. So ion exchange chromatography is applicable for the separation of charged molecules. As the name suggests, ion means it will be charged, right? Now in this technique what will happen, the stationary phase that will be solid insoluble matrix and it will be covalently attached with ions. It can be either anions or cations. It depends on the exchanger. I will tell you later what is exchanger. Now the mobile phase will be liquid phase and that will be attracted to the ion exchanger by electrostatic force. So what is ion exchanger? Actually ion exchanger can be two types, cation or anion. What is ion exchanger? It will be easily it can be easily said by these uh, examples, that is the two types, cation exchanger and anion exchanger. Cation exchanger means it will exchange the cation. I will tell you later in with the help of diagram. It will exchange cation, and anion exchanger means it will exchange the anion, right? So for cation separation we can use cation exchanger, and for anion that is anion exchanger. Okay. Or it is also called, there is another name, it is called acidic ion exchanger, and this is called basic ion exchanger or anion exchanger. Okay. Now what happens, each type of exchanger is classified as strong or weak also. Okay. Now the main thing is in the diagram. I mean the main idea you can get from the following diagram. So let's check the diagram. In this diagram you can see some columns, okay, and you can see the plus sign. This denotes a cation. Okay. Now there is the matrix, insoluble matrix, and that is attached with cations. Okay. So it will be an anion exchanger. Why? I am telling you why. So now the mixture of solutes will be introduced in the column. So there will be plus, there will be minus, everything. But you know, only the anions will be attached to the stationary phase, because the stationary phase is positive now, right? But the cations will be free, right? So with the time, what is happening here, so anion is getting attracted to the stationary phase. So that's why this is an example of, this is an drawing example of anion exchanger. Similarly for the cation exchanger, this plus will be minus and it will attract the plus. So those would have been, that could be a cation exchanger, right? So eventually what will happen, we can get the positives, right? That's why it is anion exchanger, is an example of anion exchanger. Now this is a general idea about the ions, but what about the proteins? So what type of exchanger you will use for the purification of a protein? Now that depends on the pi of the protein. Let's say uh, the pH of the solute is greater than pi. So it will what happen, the net charge will be negative. So you have to use, or we have to use the anion exchanger. Okay. So similarly if pH is less than pi, the net charge will be positive. So then we will use cation exchanger. Now the thing is, when pH is equal to pi, so what will happen? There is no net charge, zero net charge, right? That means no binding to the ion exchanger. So if the pH of the solvent is greater than pi, so you have to use the anion exchanger. If the pH of the solvent is less than pi of the protein, then you have to use the cation exchanger, right? Now there are so many examples of cation exchanger and ion exchangers, right? For anion exchangers, like some example is DEA, DEA cellulose or Q saccharose, and there can be also classified as strong and weak, as I said. Like DEA cellulose is uh, weak, Q saccharose is strong. Okay, for cation exchanger the examples are like CM cellulose, this is weak, or SP saccharose, that is strong. And remember, the cation exchanger is acidic and anion exchanger is basic. It can be also weak anion exchanger, strong anion exchanger for cation, also weak cation exchanger, strong cation exchanger. So weak cation exchanger, this is the spelling is wrong. Example is CM cellulose, strong is SP saccharose, CM cellulose. DAE cellulose is anion weak exchanger, and Q saccharose is anion strong exchanger. Okay, so this is how we will choose what type of ion exchanger you will take for your purification of the protein. Suppose if uh, pi of any protein is 3, okay, and the solvent of the the pH of the solvent is greater than 3, so then what will you use? pH is greater than pi, so you have to use anion exchanger. Okay, and if the reverse is having the pi is 3 and pH is less than 3, you have to use the cation exchanger. Okay, so I hope you have understood the general idea of ion exchange chromatography. Okay, so see you on the next video. Thank you.